So I just want to say what a pleasure it is to be at this conference amid like-minded people. I really resonated with a lot of the comments that the speakers yesterday voiced. And I'm going to spend a little less time today persuading everyone that you need to do naturalistic modes of behavior, because I think the case has already been made and we all sort of believe that. That's why we're here. In my lab, I come from a tradition of vision in a sort of natural in the traditional sense, where we study receptive fields and the V's, V1, V2, V3, V4. Lately, my lab has been interested in social aspects of vision. And I'm going to ask a relatively straightforward question today that's very much in the spirit of this conference. Uh-oh, I already have a problem. Unfortunately, my computer is, oh, here we go. Ah, thank goodness. And that is, as we develop concepts using paradigms that are rather controlled and in the mode of having sort of tight experiments, how well do the concepts that we derive from these paradigms extend to more natural modes of visual behavior? So what I mean is, if we look back on the history of visual neuroscience in the last, say, 50 years or so, there's really been an emphasis on making sure that the stimulus you present is in the right part of visual space. You have an animal either with its, eyes paralyzed or fixating a small spot on the screen, or you have a human having a fixation task. And usually you are looking at individual stimuli that are presented in an isolated and brief mode onto the screen. And that's very different from the conditions under which our visual system evolved. So you can sort of contrast that with any of a number of more naturalistic scenes. What I'm going to be talking about today is what happens if an animal is looking at a scene like this, but you can just as well take it a step further and say, what if the animal is the animal in this scene and is experiencing the immersion in the real world and the natural geometry of vision and all of these self movements and the resulting consequences. So I'm taking here the baby step approach, and I'll describe what I mean by that. The talk is first, before getting to the outline of the talk, I just want to acknowledge the main postdocs that have been involved in this work, particularly highlighting Soo-Hyun Park, who's really driven the part that I'm going to be featuring today, which is the combination of the single unit and fMRI recordings and the naturalistic videos. Okay, so I'm first going to give a bit of a background, and sort of contextualize how we got to where we are now. Then I'm going to just give some examples of what it's like when you're tracking activity in the brain as a macaque monkey is viewing naturalistic movie type stimuli. Then I'm going to talk about a method that we've developed that we believe allows us to go a bit further than we have before, by using the readout of the brain as a way of characterizing the activity of individual neurons. We call that single unit fMRI mapping. And I'll try to, at the end, draw some conclusions from our results. So it's clear that vision scientists think about vision from a particular perspective. And I just want to point out it's really only one perspective, but it's nicely summarized in this diagram that Simon Thorpe and Michelle Faber-Thorpe put together in 20 years ago. And it's the fact that it's 20 years old is not a problem, because our field conceptually moves rather slowly, I would say. Basically, vision scientists sort of think about how this process works as information sort of starts in the retina, progresses through the thalamic relay into the primary visual cortex, at which point you have retinotopic representations that come at certain latencies. Then there's a set of filters or transformations that allow you to have higher and higher representations or modes of encoding. And then ultimately, why do we do vision? Well, it has to have some effect on our behavior. So then we can try to complete the circuit by having information, again, sort of flowing through a series of stages in a hierarchical fashion that ultimately allow you to move your finger. Now, this diagram was meant to make one particular point from one particular paper it was highlighting. But this is in some ways, I think, it captures a lot of the sort of concepts that people in our field have developed over the years, and based on very good and careful and systematic work, I should say. Looking at some of the higher level visual processing areas, if you get to the sort of midway through that trajectory I just outlined, back as early as the late 1960s, Charlie Gross and colleagues, by doing single unit recordings of anesthetized animals, recognized that cells there and their visual responses were not just selected for dots of light or moving patterns, but for shapes that seemed increasingly to have biological significance. And it's worth just reflecting that in the beginning, that seemed ludicrous. It seemed like wishful thinking, and that couldn't possibly be how the brain worked. And the idea that there were hand cells and face cells was very controversial and disbelieved by the hardcore visual physiologists for decades. It continued to sort of be a finding. People would find evidence, and it was never crystal clear, but increasingly it seemed like not only were some cells kind of designated for high-level stimuli like faces, but even individual faces. Dave Parrott and colleagues did an enormous amount of very ahead of its time work. But before there were very fancy 3D computer renderings, they would actually have people posing in front of the monkey and doing various bodily gestures and hand gestures, just trying to understand what were the important things that these temporal cortex neurons were selectively responding to. And really, as early as the early 90s, there was a pretty good idea of what the spatial layout of face selective neurons was in the temporal cortex of the macaque monkey, as captured in this review. And then with the advent in the late 90s of fMRI, it became more straightforward to do mapping of these kinds of networks, including face networks. So Nancy Canwish was really a pioneer in this. And so the fMRI experiment, which is still the type that we use today in my lab, is you have blocks of faces and blocks of other objects, and you just contrast the time courses of voxels in different parts of the brain. You get a map which voxels across the brain respond more to faces than other objects. And when you do that experiment, increasingly with better and better technology, you find that in humans and in the macaque and in even the very small marmoset monkey, you have networks of these face selective areas, we call them patches now, that are circumscribed and seem to be discrete and eerily similar in the different species. And I should just point out quickly, it didn't really have to be this way. I mean, the human brain is about 1,400 grams, and the marmoset brain is about 8 grams. So that's a factor of almost 200. But nonetheless, this aspect of brain organization seems to be, at least at a superficial level shown here, preserved. So these are areas that respond more to faces than to other objects. Doris Tsao and Vinrik Freiwald went further, and they said, what if we use this to target areas and we direct our electrodes there to one of the sweet spots? What do we find? And what they find is a very high fraction of neurons that respond selectively to faces. We've done the same thing subsequently. When we do it, I'm just going to show you results from these highlighted face patches. The specific names are not important. But if we go to the ML, which is the middle lateral face patch, and we direct these microwire bundles that allow us to have a very dense sampling of cells simultaneously recorded within a face patch, we can target them. This is the MRI shown here. And we can say, are the cells here responsive to faces selectively? This is an example of one cell. If you show a bunch of faces, the response in the following the vertical line, the red response is a response to faces, the blue to non-faces, and clearly that cell likes faces. If you look across the population, my field has sort of adopted this mode of showing things in terms of heat maps, which I think is a nice way of summarizing data. Here you have 40 or 35 cells that are in order in terms of their selectiveness for faces, but everything to the left of the vertical white line are face stimuli, and every row is a neuron, and you find that the majority of cells like to respond more strongly to faces, indicated by the yellow, than they do to non-face objects, more in the blue tones. And if you do that for these other face selective areas, you find something similar. That's just basically qualitatively similar for these 3 different face patches. I'm not showing here what happens if you go between the face patches, but you don't find this between the face patches. So there are it's reflecting what you find in the fMRI. You find face selective cells and face patches for briefly presented face stimuli. And this is very much in the tradition of visual neurophysiology that's led to the model I mentioned before, where you have an animal, in this case, looking at a small fixation point and briefly presenting stimuli of different sorts on the screen. And this is every lab uses some version of this, and it's a little bit in contrast to what I'm going to feature today, but we certainly indulge in this a lot. I do want to just point out that it kind of draws you into a certain way of thinking, and I think over the decades it's drawn our field into a certain way of thinking. So if you have a bunch of different stimuli and you present them one after another, and then you find that you're recording from a cell and you find that the cell responds more to one stimulus than to another stimulus, you start to kind of say that that cell is representing that stimulus, or somehow the stimulus is encoded by the response to that cell or to multiple cells. And then you have to have an area that's reading out this encoded information. Now, I'm not, this is never going to be wrong, but I just think it promotes a certain way of thinking that's very much tied to our analysis methods. That's just one perspective on the brain. I don't think it's that compatible with the more free-running type of continuous brain activity you have under natural conditions. So I think I want to sort of, in my own lab, we sort of try to push back on this notion that everything is first encoded and then decoded in this sort of serial manner I showed before. You'll see a bit more what I mean by that in a moment. So within this framework, as my lab often operates, what do face cells encode? Well, one thing is identity, and so I won't spend much time on this, but just to give you sort of an example. So, Wienrich, Freiwald, and Doris Tsao and Mark Livingstone have looked at the way that cells in the face patches respond to the parametric variation of different features in cartoon faces. Doris Tsao's lab and my lab have looked at the effects of morphing according to sort of what's called a face space, and seeing parametric variation of cells in the face patches. And if you contrast different areas, different face patches, and have not just identity, but other things like, rotational transformation, you can start to see what appears to be a progression, maybe a hierarchical progression of processing from one face patch to another face patch. And you can begin to do what we like to do as scientists, which is to think about like how that, mechanistically, how are the operations in that hierarchy progressing from one step to the next step, usually from posterior to anterior. Now, if you kind of put that together and just think a little bit more broadly about the principles from systems neuroscience, one principle, and this is what's going to come up in the talk today, is functional segregation. So I just showed you, and it seems to be true for face patches, that neurons that are really within the same region have similar roles. In this case, they're responding selectively to faces compared to other stimuli. Of course, you're loading the deck a little bit because you have a very contrived stimulus presentation paradigm, but that's, it seems to be organized in that way. Then there's the notion of hierarchical processing, and there's a lot to say about that. You can sort of track the latencies as you move away from the retina. You can also look at the anatomical connection principles and other variables that are sort of compellingly given an idea that there's some hierarchy in processing, although that's often from the perspective of a sensory input, which is only, again, one way of thinking about how the brain is organized. And then there's domain specificity, which is essentially assuming or believing that the brain has dedicated modules that are really dedicated to singular operations, like face patches and body patches and color patches and scene patches and things like that. Again, these can't ever be completely wrong, but I think the extent that we're invested in these concepts is very much shaped by our methods. And I think one underlying assumption that's not often enough stated is that there needs to be some capacity for generalizing. So if we take a lot of experiments from reduced paradigms and generate concepts over decades, that should have some bearing on normal brain function. And if it doesn't, we're in quite a bit of trouble. So today, like my real goal is to let's just look at something that seems to be pretty solid. The face cells and the face patches, they respond to faces, and see to what extent can we find that to generalize in just a little baby step away, where now the monkey is just sitting there watching videos. So I'm going to begin by just saying, well, what does the brain do while the monkey is watching videos? And I should just say that this work was very much inspired by one of the attendees, which is Uri Hassan, who we'll hear from later today. And we really sort of copied his mode of interrogating the brain, and hopefully extended it a little bit as well. So the fMRI and single unit responses from naturalistic videos, if we just look at fMRI, which is where Uri started, let me just say a little bit about the fMRI that we use. This is a vertical scanner that we have at NIH. We've had it for a long time now, like 20 years. And the monkey goes up into the vertical bore and looks at the movies through a mirror onto a screen above its head where it's projected. And then we're going to be contrasting that later with the single unit recordings from the face patches using these microwire electrodes. And I just wanted to mention from the onset that the microwire bundle is full of electrodes that are very localized. So we'll be recording from 50 neurons at a time that are all within 1 millimeter, or you can think of all within one voxel. So we're looking at local populations of cells. Okay, so if we go back now, and we're talking about the tracking of fMRI signals to the viewing of the movie. So we had 15-minute video footage of macaques and other primate species doing interesting things. This movie was selected and curated by Brian Russ when he was a postdoc in the lab, mostly just to keep the animals interested. The animals were allowed to freely view the events of the movie. We tracked their eye movements, but we didn't make them fixate, so they have a different viewing pattern each time, a somewhat different viewing pattern each time. And if we just want to get a feel for what is the fMRI set of responses during the viewing of the movies, this gives you a bit of a feel for that. This is now sped up because the whole process is rather slow. But as we move forward with the video here, and just kind of going from scene to scene, the left shows the hemodynamic response in the sagittal slice of a monkey watching the video. Exactly how we did this is not that important, but you can see there's a sort of churning, some synchronous fluctuations, some that are a little bit asynchronous, with voxels having some level of independence in their time courses and some level of covariation with their neighbors. So you can try to look into the movie and say, well, when does the voxel go up and when does it go down in its activity, and do what visual neuroscientists normally try to do. But we quickly abandoned that. I'll show you a lot of alternatives to that today. But one real solace that we took was that if you look at multiple presentations of the same video, this is now with our, unfortunately I don't have a pointer, but with our Mayan signals, we have very high signal to noise. We can see that the different colors in the trace there from the single voxel in V4 is very reliable across trials. So multiple presentations, this is for one 5-minute movie, multiple presentations of the same movie led to responses from this voxel, which was a representative voxel, that were very reliable across conditions. So that means that despite the fact that the eye positions are changing somewhat as the monkey sees the video multiple times, the voxel time course is quite reliable. So that allows us to have good confidence that at least we may not be able to know exactly why it fluctuates in the way that it does at each moment in time, but at least it knows what it's doing and is driven by the visual content of the movie. Okay, so one thing we can ask is it possible to do something that's kind of like a normal visual mapping experiment? So that's how we started. So if we have voxel time courses at different positions, I'm just looking at a couple of different voxels in STS. Here's one time course with different runs from the same voxel. A run is just a presentation of the movie. Here's another voxel, here's another voxel. So within the same vicinity, you can see there's some variation in the voxel time courses, and that's related to the functional differentiation within this area. is it possible to make maps of the relationship to different visual parameters? Now we're saying, how does it link to the visual structure, which is in a sense the traditional question that you ask if you're a vision scientist. So we can do that by just looking at each voxel's time course, and we can in parallel look at the features of the movie as they're evolving along the same time course and say which of the feature content seems to map the variation in a given functional voxel. And how we did this exactly is explained in the paper listed below. But we can essentially look at motion or contrast or the occurrence of faces. You can compute all of these things in many different ways. And you can say what accounts for the highest amount of variance in the voxel that you're tracking its fluctuations over enough time so that you're out of the range of spurious correlations. Now, when we did this, we asked a rather straightforward question, which is, can we map face patches in the traditional way I showed before, but now using just a free viewing video? The monkey's not trained, there's no fixation, nothing like that. By just having the monkey enjoy a movie, can we map face patches? And so when we did that, this is 3 slices from one monkey. What's shown here are just the traditional face patches that are mapped with a usual block design. So this is what you get per default that Doris Tsao showed for the first time and many, many labs use now. If we do the same thing with the monkey watching the video, what we found was that it really looks very similar. So now we are extracting the faces as they evolve over time in the video. We're taking that time course, we're mapping it back onto all of the voxels across the brain. And what's shown in orange are the voxels that are correlated significantly with that time course. Again, this is something that's, again, very similar to what Uri had done in his initial studies a while ago. And in the second monkey, it looks very similar. So yes, so far the story is pretty simple still. I mean, you have these face patches, they have face cells inside them, and then if you even look in the movie, you can find that the face patches are correlated with the occurrence of faces in the movie. So that makes it sound like, yes, we can step outside of contrived paradigms and see a recognizable functional differentiation even with naturalistic viewing. I'm afraid that's where the good news really ends. Then it gets more complicated. When we look a little bit more closely, we notice that the face content that was giving the exclusive identification of the face patches that matched so nicely the contrived paradigm only accounted for a small proportion of the explained variance of the voxel time courses. When you think about it, the movie is very rich now. It has dimensions that are not present in most of the previous face mapping experiments. It has motion and geometry and various other kinds of things. So the explained variance, so each of these panels corresponds to the response for one face patch, including the prefrontal face patches, which are the P. The fact that there's any green there, which I'm just showing the faces now only, is the only thing that qualifies it to be identified as a face patch, to show up as an orange voxel in the previous slide. But if we compare to the other attributes within the video, we find that the face proportion of variance displayed is very small, and it's actually in almost all cases smaller, actually in all cases smaller than the amount captured by motion, which was computed completely independently of any faces. So this sort of, I don't mean to make a big point of this, but it sort of raised our awareness that, oh yeah, wait a minute, with videos, there's so much extra stuff that maybe we're missing something, and it's still possible that you have these exclusive areas that are uniquely, in some sense specialized for faces, but that's just the tip of the iceberg in terms of what they're doing under more natural viewing conditions. So let me just summarize that quick point about the fMRI first. So voxel time courses are highly repeatable. There's an accurate mapping of face patches, surprisingly accurate, despite the fact it's a very different paradigm. However, the explained variation in the voxel time course during the video viewing it's exclusively restricted based on the faces, but that even within those areas, it's not much responding to the face content. And it was much more dominated by visual movement in a way that wasn't very different from adjacent, just outside the face patch, it was also in a similar way dominated by visual movement. So there are different operations that seem to be overlain within the same cortical territories. Okay, well, what happens if we then target these individual face patches and look at face selective cells? I'm only going to be talking really today about face selective cells. You find some that are not face selective, but we've excluded them from the analysis. Let me give a feel for what that is like. And to the extent that you'll be able to hear this, you have to have a look at this video and then listen to the cell. And this is selected because it's a typical face selective neuron when you're trying to see it in the context of a natural video. So I don't know if you could hear that or not, but if you could, it's a little frustrating because it's a little hard to pin down exactly what it was responding to. And it seemed to be related to some things about the movement of the animal. Sometimes faces appeared, it would go. Sometimes faces would appear and you wouldn't hear much. That is what it's like. So I've sat and listened to hours of this, tried to just get an intuition for what the face cells respond to. And I have to say, it's really inscrutable. It's very hard to determine. One can't rule out it's a combination of features. It has to, at some level of description, be a combination of features, but it's not an obvious thing that pops out that you can hang a name on. Like every time a face appears, the cell responds. And it gives you the impression it's much more than faces too. It's like faces conjoined with other things that are related above. I think one thing that's underappreciated is the geometry, the geometric layout of the scene. I think plays a big role in this. But again, so what are we going to do with that? Well, one thing we can say is how repeatable are the responses across trials, and that's where we have again a little bit of breathing room, because if we give the same stimulus across different trials, I think this is for a few minutes. Now 10 different trials, each of the little tick marks is a raster. Each row is the presentation of that same video on a different trial. And what you find is that there's a video-driven sort of synchrony across trials. That means that the cell is responding to the same events in the video, again, as the monkey in this case is freely viewing. So that means in the same way we were talking about that with the fMRI, we can use the electrophysiology as a sort of reliable measure. Okay, so let's then ask the question, okay, if we have face selective cells, they're pretty similar in at least at a categorical level in what they respond to. Are the responses to the video also similar from adjacent cells? Again, they're all within a millimeter here. So this is some work that Kenji Kayano collected, asking this question. Now, so first the monkey watches the flashed faces, and that's what's shown here, faces and other objects. Again, my pointer is not working, but the top row here, you can see a color table. Sorry, it's a different color table than before. Now, orange is responding to faces, and cyan is not responding to the other types of objects and scene stimuli. These are ranked. So this is the most face selective cell. If we look across the population of simultaneously recorded neurons for this particular set of recordings, which is in the AF face patch, there were 58 cells, and maybe, at least a half, maybe 2/3 of them were face selective. Then we say, okay, if we kind of have a look at that in a traditional way, so that cell responds more to faces than to the other object categories. And if we go down and just select a few of these cells, kind of randomly, really, these are all, by anybody's definition, face selective cells, and that they're responding in a categorical way. And we say, okay, these face selective cells, all in the same face patch, in fact, all within 1 millimeter, how do they respond to the events of the video? And so if we now have the monkey watching the video instead, these are the same cells that are being recorded for the video now. So here's one response from one of the cells, just to illustrate. Again, what you can notice is that across 10 different viewings, the cell is doing the same thing. So again, it has that sort of reliability. It's not doing random things. But then if you go to its neighbor and its other neighbor of face selective cells, as I go from one to the other, all simultaneously recorded, what you find, and I'll put a little guidance on here to help you see the patterns, is that the temporal structure of these, while even though it's very reliable across trials, it's very different from its neighbors. So there's a lot of variation in temporal structure. And so already you kind of have at this level, it's still sort of impressions. The impression you have if you flash different images is that the cells in the top half of the figure are kind of all doing the same thing with some variation. And if you look at the left half, they're all doing very different things and are largely uncorrelated. In fact, this distribution here, this is how reliable it is across trials for an individual cell. So it approaches 1. If you then say how reliable or how similar are cells that are right next to each other, that's this distribution. So the correlation value of the time courses of the cells to the movies are, it's the mode of the distribution is about 0.1. It's got long tails. You have some cells that are similar and some that are anticorrelated, but it's very much the case that adjacent cells are doing very different things from one another, even though they're very reliable across multiple presentations of the same video. So let me just summarize that part before we get to the exciting part. I really have no idea how much time I have, but spiking time courses are highly repeatable. However, that's kind of the only piece of good news. It's really hard to identify what is what and what the cells are responding to in the video, particularly if you want to assign a name to what kind of cell this is. It gets really almost impossible, I would say. Neighboring face cells respond very differently to the same content. And this has got us thinking, so where do we go from here? And so the second half of my talk, I might have to abbreviate a little bit of it, is asking, are there some alternative ways to characterize visual neurons that don't involve a direct reference to the visual content, which might sound counterintuitive, but that visual content, you either have to sort of have words or you have to parameterize it or something like that. So can we do it in a different way? So that's what I'm going to explore. And I'm going to start by asking, why could it be that adjacent neurons that are so close to each other respond so differently? And like here are a couple cells and here are the time courses, and they're now, you can tell that they're repeated across multiple viewings, they're reliable. So why they'd be so different? Well, one possibility might be related to the laminar structure within a cortical column. And you could say that cells that are within a column, they have a distribution of labor so that it might be unknowable to us, but they have different roles, and that's why they seem so different. Another possibility, which is not really mutually exclusive, but one that I think is more favored, at least by me, is that cells have some shared input, particularly from inhibitory components of the local microcircuit, but they also have some private connections from long-range inputs from all over the place. And this selective afference, I think, is going to be expressed in a different way according to whether you're flashing images or whether the animal's engaged in a rich, dynamic video type stimulus. And so I think this enhancement of these different inputs might be one of the reasons for why we see such a diversity when we have the video stimuli. So can we kind of get to that? Is there some trick by which we can tap into that? And that's where the whole brain fMRI comes in as a tool that we've tried to harness here. So the last part I'm going to tell you about is the single unit fMRI mapping of face patch neurons. So here, the idea is that you have one monkey viewing the video while you're recording from single units. In fact, we had 11 monkeys doing this. And then another monkey viewing the video in the MRI, and we had 2 monkeys for this. And they don't even have to be the same monkey. We've done simultaneous things too, but I'm not going to talk about that today. But what you have is a time course that serves as a sort of common denominator. And so you can take the time course of individual cells recorded from face patches in this case, but it doesn't have to be. And then you can say, how does their time course, the video-driven variation, match that of voxels all over the brain. And that gives you a transformation of sorts, or a readout for what individual cells are doing. So you're not really talking about, you're not trying to characterize cells based upon the video content or the visual preferences. You're trying to say this cell is doing the same as these areas, but not those areas, or it's some link to it, or maybe you say it participates in the network, or we don't quite know how to talk about it yet. But it allows you to classify neurons in a new way, which is based upon their covariation with the whole brain. And it's not particularly arduous because you don't have to have even simultaneous recordings inside the scanner or something like that. You just have to have a set of data collected from video viewing with electrophysiology and a set of data collected with the same video with fMRI. Okay, so I'll just quickly go through this. The details are not that important. I think you probably already get the point, but you have a bunch of different cells. They all have their own time course. Then you have a bunch of voxels across the brain from your readout data set. So that's your fMRI readout data set. You just, you have to take care of hemodynamic shifts and smoothing and things like that. But you can then look at the correspondence at each voxel across the brain and make a correlation map. So one cell might give you a correlation map that looks like this, and I'll go into great detail on this. Other cell might give you a rather different correlation map. So each cell now has a map that characterizes it. And that's what's the shared video-driven response for that cell across the brain in the terms of a correlation map. So if we look at one cell, this is a cell from the anterior medial face patch. And this is just, again, I'm only talking about face selective cells for this whole talk. And we say, what is the map that corresponds to the temporal variation of this cell in response to the movies? So first of all, here's the face selectivity of that cell, just again to show you that it's face selective. So now, this is the answer to that question. So I'm just showing the cortex for this talk. The recording site is one cell within this green area here, the face patch AM. And you're tracking its fluctuations as the monkey watches the movie. And if you say what co-fluctuates with that single cell, it's what's shown here in orange. So these are the areas where there's a selective coupling with that individual cell. And as I've told you, neighboring cells are doing very different things, and I'll show that in a moment. But this is what it looks like. Now this map, first of all, you can also say it's bilaterally symmetrical. It's not functional connectivity. It's a different thing. It's a video driven, and these are responding to the same visual features. The cell is as the responses in these voxels that are orange. Now this kind of looks reasonable. Let me just flatten it so you can see it in a different way, because that's how I'm going to present the next few slides. So if we take that same thing I just showed you there and I just flatten it into a cortical flat map, it looks like that. The other hemisphere looks like that. Now we can take from the same monkeys, we've also collected other things, including the face patches from more conventional testing. And if we overlay the face patches, we find that, oh, for this cell in the AM face patch, this cell covaries only exclusively, but not, in its own way, with the voxel time courses of the voxels in the other face patches. So this cell is linked exclusively with the activity in other face patches. So that's kind of interesting, and that might even be your first guess. If we look in other face patches, let me just say our recordings now, we exhaustively looked with a lot of monkeys in 4 different face patches, and we wanted to say, so how diverse or how uniform are they, and how much is there a progression across face patches? So that cell I just showed you, now that's a map that represents the responses of a cell. That's the one I just showed you. Now, if we look in these other face patches that are as far as a centimeter away in a monkey brain, which is a long distance, you find that kind of cell is present in all 4 of those face patches. It's not very common in any of those face patches, but you find examples of it in all 4 of those face patches. So that means that it's not unique to one of the face patches. And then you find also, and these are the recording locations again shown here, and so you can sort of summarize that. Okay, this is sort of a mode of activity that's shared that's related to these face patches. Now, here, the top row is what I just showed you, but I think one of the really important points is that if you look in the same face patch, I'm just going to try to finish up quickly here, you find that a neighboring cell will have a very different pattern of activity. And that different pattern activity is also shared across the face patches. And then a neighboring cell will have a very different pattern of activity, in this case, related to central V1. And that's also shared. And another pattern of activity that's also shared. And you keep finding that the idiosyncratic patterns that differentiate neighboring cells so starkly within individual face patches is actually quite shared across the face patches. So I think in the name of time, I'm not going to take you through the analysis details. We had some technical problems in the beginning. Let me just go to the punchline here, which is that you find these groups of neurons where each group here, which is shown in the central portion of the figure, these are arranged by regions of interest. You'll have to take my word for it that we did this in a systematic way. The neurons arrange themselves into groups, and the groups are the areas that they come from, are indicated by the coloration of that bar on the right-hand side of the middle pattern. Okay, so group 1 draws from 3 different areas. Group 2 draws from 4 different areas, and their shared mode of covariation is reflected in the maps that are shown adjacent. So I can't point, but group 1 in the upper right, that seems to be linked with a subset of face patches and orbital cortex. Group 2 is anti-correlated with early visual cortex and a few other items in the STS. Group 3 is very strongly positively correlated with activity throughout the STS, not just face patches, etc. And so you have about 6 different functional species of neurons that are all shared throughout the different face patches. And if you look across the population, the maps on the left here are showing the distribution of these modes, and the right shows the prevalence of these different functional types within the different face patches, which are enumerated on the top. So in other words, you have broadly shared rather than individual operations that are restricted to individual face patches. So let me just summarize what we have learned from this. So first of all, as other speakers have said, and probably it'll be said more often, having some naturalistic perspective changes things a bit. So neurons, first of all, they respond reliably to naturalistic stimuli in our hands, which was really useful because it was somewhat constrained. I mean, it wasn't that the monkeys were completely free. They were still watching a video repeatedly. Neighboring cells had very diverse responses, and it wasn't possible subjectively to make much headway in terms of understanding the response properties of individual cells. So the impression that you have for flashed images might be what you have here on the left, is sort of different shades of the same thing. Maybe it's all face selective, but some respond more to some faces and some more to other faces. But the impression you have when you have the naturalistic videos is that the cells are just doing completely different visual operations. If you look at the whole brain readout, this is a method that I think is going to be very useful. It's not better. It's just a complementary way of evaluating complex data from individual cells. It's very data driven. It doesn't force you to make decisions about trigger features or names, or you don't have to load the deck too much in terms of your experiments. And it really drew our attention to, which we're interested in now, why are these certain coalitions of areas that we're going to tentatively call networks because we can't think of a better word right now. Why are these the ones that are going together in these 6 different modes with the cells in the different brain areas? These are the ones that I meant to refer to right here. And it gets us to think about networks in a different way as well. So for the face network, which is sort of one that has been studied in great detail, here's how we're starting to think about it. There's a local intermixing of functional types of neurons, and there's a similar kind of mixture across different face patches. And we're talking about face cells. These are the face selective neurons exclusively. So it seems that everywhere locally you have this mixture, but it's kind of similar elsewhere. There are different proportions, but there's representation of each type in each face patch. And we know from Doris Tsau's work that the face patches are very strongly interconnected. And so we're sort of speculating that like species of functional neurons are interconnected with similar species in other face patches. And so the organization of, if there is some kind of processing hierarchy or drawing together of different kinds of operations, it doesn't seem that it's from one face patch to the next stage. It seems that it's much of a more parallel set of subnetworks than that. And the wider implications of that, first, I think, as has been mentioned yesterday as well, it just like when you try to say, what is the organization of the brain? I mean, okay, the moment you're talking about functional organization, it just looks different under natural behavior. This was really a surprise to us. It just seems that the level of homogeneity is expressed very differently. Areas appear less domain specific and less functionally segregated. And this, I think it's exciting in some ways, but it really poses challenges for a lot of neuroscience methods that really rely upon or assume a strict segregation of function within the brain. And I think the punchline maybe of this whole conference is it's really critical to constantly test the validity of the concepts that we get when we just move outside of our reductionistic paradigms, because otherwise it's really, what's the point? I mean, we're really needing to get to this thing about how does the brain operate under normal conditions. And I'm just going to end the talk there. And thank you for your attention, and thank my many collaborators and trainees who have been contributing to this work over the years.